Welcome to the latest edition of the Mind-Gut Conversation podcast, a place to learn about breakthroughs in the science and practice of health, mind-body interactions, the microbiome, food, and the environment. Today, I have the great pleasure to talk to Dr. Colin Hill, professor in the School of Microbiology at the University College Cork, Ireland, and principal investigator in Elementary Pharmacology Center, Microbiome, Ireland, one of the leading microbiome scientists in the world. Colin's main interests are in studying the impact of the human microbiome on health and disease, and particularly the role of bacterial viruses or so-called bacteriophages. He has published more than 650 peer-reviewed papers and holds more than 25 patents. Colin is a member of the Royal Irish Academy, the highest honor for an Irish academic. In 2010, he was elected to the American Academy of Microbiology and was awarded the Metchnikoff Prize in Microbiology. Last but not least, he served as president of ISAP, the International Scientific Association for Prebiotics and Probiotics, from 2012 to 2015. Welcome to the show, Colin. I'm really excited to talk to you about a topic that neither myself nor a big part of our audience will know too much about. And I want to start with the observations. So all of us have experienced and read and had saturating information about a particular virus, COVID-19, that has basically changed the world and the way we interact. So most people will base their view of viruses on this type of virus, something dangerous, something coming from the outside, something causing diseases. And but what your area of interest is really those viruses that live inside of us, obviously not dangerous and probably beneficial. Do you want to expand a little bit on that?
Sure. Let me start by just saying something about viruses in general. COVID-19 has really shaped our world over the last 2 years, but viruses have shaped our world, the microbial world and the human and the animal world, plant world, since evolution began. And viruses are very odd creatures. They're absolutely beautiful. I mean, people have seen probably these lovely drawings of COVID-19 that are in all the newspapers and websites, but they're incredibly diverse and very pretty little structures. But they fall somewhere on the border between life and non-life, if you like. They replicate and they evolve. So in that sense, they're living organisms evolving here on the planet. But they're obligate intercellular parasites. They can only live if you give them a living cell in which to replicate. And the fate of the poor cell that gets infected is not a good one. So if a virus infects a cell, that cell is likely to die. Not in all cases, but generally it's likely to die. And if you're a human being like us and you have billions of cells, and you can afford to lose a few and the viruses replicate and eventually the immune system will take care of them. But imagine if you're a bacterium and you're only a single cell. Then if you get infected by a virus, there's a life or death outcome for the bacterium. Either the bacterium wins and fights off the virus, or the virus wins and the cell is killed. So at the level of the bacteria that live within our gut, viruses present a significant threat at the level of individual cells. And they can probably then shape communities in that way. A virus is the most powerful replicating machine on Earth. There are more viruses on Earth than any other biological entity. It's estimated there are about 10 to the 31 viruses on Earth, which is a number you can't even conceive of. And the reason there's so many is that, for example, when a bacterial virus that we call bacteriophages, when they infect a bacterial cell, within 20 minutes, they might produce 100 to 200 bacterial or bacteriophage particles. Now, a bacterium in half an hour or so can only double. And so a bacterium can go from 1 to 2, a bacteriophage can go from 1 to 200. So they can rapidly kind of burst through a community of bacteria, destroying the bacteria, liberating millions of copies of the virus, and having an impact then on the bacteriome, the bacterial part of our microbiomes, all the microbes that live within us. And so in a strange way, these viruses, they're not dangerous to us in any way, but they shape the bacteria within us that influence our health. So in that way, the bacteria can have an impact on health essentially at one remove by influencing bacterial function and bacterial populations.
I mean, that's amazing to hear, because I mean, all the interest and the, the research interest and research investment and progress has really been made on the bacterial side. I mean, we think when you listen to some of the, on some of the podcasts and some of the information in the media, but also in the scientific press, it seems like we don't really care about the viruses. We seem to know almost everything now that we need to know for to understand diseases and how to treat diseases and diagnose diseases. So based on what you say, that's far from...
Well, that's one of my big bugbears, and I'm fighting a losing battle that, people just refer to the microbiome. And really what they're talking about is the bacteriome. They're almost always just talking about the bacteria. They're leaving out the archaeome, which is the archaea, the kind of an ancient form of bacteria that we have a lot of in our gut. They leave out the molds, the yeast, the fungi, they leave out the viruses, but yet they just call it the microbiome. And I think if we do that, language is very important, because if we just simply dismiss all the others verbally, we kind of dismiss them mentally as well, and we don't want to think about them. The microbiome is already complex enough without having to engage with the other aspects, the non-bacterial aspects of it. So people, and I've done it for years, shy away a little bit from that. But I really believe that, the bacteriophages, the bacterial viruses, they're the predators in this environment. It's been estimated that half of all the bacteria on Earth are killed by bacteriophages. So if you're a bacterium, one of the most important things in your life, and potentially the cause of your death, is bacteriophages. So you're going to be constantly aware of them, trying to avoid them, evade them, defeat them. And so to try and study bacteria in the absence of this force, I think we're missing a trick. I use an analogy sometimes with students, and I know analogies can be, can be troublesome at times, but in Australia in the late 19th century, they released something like 200 rabbits into Australia, and rabbits have no predators in Australia. And so now there are billions of rabbits in Australia. And if you were an Australian scientist studying rabbits, you might wonder, why do they have such big ears? Why do they have such acute hearing? Why do they have such big eyes and good sight? Why do they live in burrows rather than just live above ground? And probably because you're a bright scientist, you'd come up with reasons for all those things. You'd say, oh, they must communicate over long distances, or maybe they have to beat the next rabbit to the next bit of grass that they want to eat. You'd come up with good reasons and you'd justify them. If you put one of those rabbits back into the US or Ireland, you'd understand within seconds why they have these features. It's because they're prey to predators, foxes, eagles, wolves, whatever. And so in a flash of light, you suddenly say, okay, now I know why they have big ears and long legs and deep burrows. And I think sometimes we study bacteria and we study what they do and how they function and what kind of things they have on their surface and how they interact with us. And we, because we're clever, we come up with reasons for all those things. But I think if you're missing this final factor, the predators, the role of predators in the bacterium's life, then we'll never really understand the role of the bacterium in human health. It's been an ecological principle for many, many years that the only way to maintain a stable, complex ecosystem is to have predators. If you've no predators, you've no stability. So I believe, and I'm trying to persuade my colleagues with varying degrees of success, that we have to study the virus. Otherwise, we're just simply looking at half the picture, and we'll never get that full understanding of how the bacteriome interacts with humans and animals and plants for that matter.
Yeah, I'm going to come back to this ecosystem concept, but let me ask you one question. So, bacteria have been around also for billions of years in the oceans, have perfected, I mean, that's one aspect that I really like, their communication system with each other, which then allowed them also to communicate with the most primitive gut, the most primitive animals, and ultimately humans. So I assume the viruses were around as well during most of that time, or all of the time. So did the bacteria ever develop a communication or a defense system against the viruses? So similar to what our our COVID-19 vaccines today are, that we came up with, quickly.
Absolutely, great question. And the bacterial genomes are actually crammed full of antiviral mechanisms because they've evolved in the presence of these viruses, these bacteriophages. And so they do so many different things. I mean, some bacteria actually commit suicide when they're infected with a phage and essentially take the phage with it. It's an altruistic kind of, look, I've been infected, I'll die and protect the rest of the population. I guess, is that a bit like people vaccinating themselves to protect the rest of the population, in not as dramatic a fashion, thankfully. Some bacteria chew up the incoming bacteriophage DNA. There have been Nobel Prizes awarded, Rich Roberts and others, for these restriction enzymes, which chew up the incoming bacteriophages. There's the CRISPR, for which a Nobel Prize is inevitably going to be awarded pretty soon. CRISPR is really a phage defense mechanism in bacteria. It's a way of recognizing that you've encountered this bacteriophage before and you need to kill it. But you can also set up a kind of a relationship with the bacteriophage. The bacteriophage can insert its genome into the bacterial genome, and they just coexist until, upon some signal, the phage decides to break that pact and burst out of the bacterium, releasing again 10 to a couple of hundred phages. And practically every year now, someone discovers a new bacteriophage defense mechanism in bacteria. But in contrast to that, the phage are still there. So to my mind, that suggests that there's more of a relationship than we might suspect. It's not just the phage trying to kill bacteria, and the bacteria are trying to defend themselves. If we think about bacteria at the single cell level, then it should be very, very afraid of a bacteriophage. But at population level, it might actually keep the bacterial population very fit. Again, to use an analogy, somewhat similar to my previous analogy, if you have antelopes in a particular area of Africa and you've lions, and you take away the lions, then maybe after a while, the antelopes will start getting a bit fat. The sick ones will not be being taken out of the population. The whole population will get less fit. Mm-hmm. Now, bring the lions back in, and individual antelopes will suffer, of course, they'll be killed. But the population of antelopes as a population will become fitter and will maintain its fitness. And also, of course, the presence of lions will stop the antelopes eating all the grass, ruining the ecosystem and disappearing themselves. So there's a benefit, albeit through a kind of a harsh interaction at the individual level, but there's significant benefit to having these predators around. So I think bacteria, in the evolutionary terms, recognize that. And so I, again, sometimes say bacteria and bacteriophage are not fighting, they're dancing. They're in a relationship, and both benefit from that relationship.
Yeah, this is really fascinating. I have to say, this whole ecosystem concept that, like initially, I still remember, and I was a victim of that myself, that we spent a lot of time talking about this, the wonderful symbiosis of the bacteria with us, and this is all harmony and nobody gets killed. But this is a very different picture, which is much more the reality, the biological reality, not our projection, our human projection onto a system. So, I mean,
yeah, this is nature red in tooth and claw. This is real nature. Yeah.
Staying with the concept of the ecosystems. So you've written about this. So there's certain mathematical equations where ecologists have characterized ecosystems, and this prey-predator relationships where you can either have oscillations that the predators increase and the prey decreases, and then it changes in the other way. I mean, one thing that's unique about the gut microbiome in general, or the bacterium, is its stability over. I mean, there's modulation, obviously, age-related to a certain degree, but essentially it's fairly stable considering the tremendous variations that it goes through. So how is this accomplished in a system with constant turnover? It would seem to me like a small change in the balance between the prey and the predator would shift the system in a certain direction. How is this stability accomplished?
I think it's accomplished in many ways. Of course, it's always more complex than the simple question, but there are physical barriers. I mean, there's, in the gut, there's mucus, there are cells, there are materials flowing through the lumen. Just because there's a phage and a bacterium present doesn't necessarily mean the phage will encounter the bacterium in a way that it can kill it. So there's literally just physical distance. The bacterium might be embedded in the mucus and a phage might be in the lumen and it can't reach it. We've also got this resistance patterns that within a like phage, one thing I should have mentioned was that bacteriophages, these bacterial viruses, they're very specific. They're very narrow spectrum. So not only will, let's say, you have Bacteroides intestinalis in your gut, there may be a phage that hits Bacteroides intestinalis, can kill it. But it may not even kill all strains of Bacteroides intestinalis. Never mind killing Bacteroides xylanosolvens or Bacteroides something else. So they're very narrow and very specific. So it's not like a phage can just spread through the gut microbiome, killing every bacteria it encounters. It can only kill those very limited bacteria which are sensitive to it. But if those bacteria increase in number and become more and more abundant, the phage finds it easier and easier to find them. So it'll kind of knock the population back on its head a little bit. It's called kill the winner dynamics. So if one bacterium in your gut suddenly finds itself in a situation where everything is perfect for it, your diet is really suiting it, it's having a wonderful time and it wants to replicate and become, overtake the population almost of other bacteria around it, then it's likely that a phage will suddenly find it easier and easier to kill it and will knock the population back to the point where it's finding it more difficult to find it again. Again, a bit like the lions and the antelopes. If the antelopes were all just locked into a pen and you put a lion in there, the lion would just kill all of them. But the fact that they have to find them first before they can kill them keeps this kind of balance. I think phage can play a very important role in maintaining that diversity and stability of the bacterium. And what we found in a study we published a couple of years ago was we took 10 individuals and followed them over a year, and we showed that their fecal bacteriomes were very stable over that year for all 10 people, but their fecal viromes were also very stable over that time. So the viruses and the bacteria are coexisting. And that's what I mean about them dancing rather than fighting. If they were fighting, you might expect either at the end of the year you'd have all phage or you'd have all bacteria, but you don't. You see them maintaining a kind of relationship with each other over extended periods of time. And some individuals we looked up to nearly 3 years, and we still find the same phage and the same bacteria present 3 years later. So I think they're a very important part of the stability and diversity that we see in the gut.
So there's been a growing literature. It's virtually, I mean, there's hardly a chronic disease where you don't have talking about the bacterium, the microbiome in its traditional sense, where there's not been reported what's referred to as a dysbiosis or an alteration in the relative abundances and diversity. So, I mean, first of all, these are all cross-sectional studies. We have no idea, about the causality of this. But do you think, based on your knowledge and your concept of this ecosystem, that the viruses could have a pretty significant role in some of these diseases and in these symbiotic states that have been observed?
Yeah, I do. But, you know, I'm falling into the trap that I think a lot of microbiome scientists fall into, of the microbiome has become a bit of a belief system rather than a scientific data-led system, where things seem so obvious that we just believe them because they're just so obvious. They must be true. And I still think we're at the point with all microbiome science, but especially with virome science, where we can generate really compelling hypotheses, I think, like that one you've just espoused, that maybe could the virome be playing a role in human health and in disease? And I think we have good grounds for doing that, but we haven't tested it rigorously yet. So I don't know what the answer to that is going to be. I hope and I believe that they are important, but I haven't proven it, and I don't think anyone else has proven it. So I think we have to leave that as an unproven hypothesis right now.
I mean, a good example for such a speculation, you know, would be Parkinson's disease, where the viral origin has been suspected, and you know, in the majority of patients, it seems to be starting in the gut, in the enteric nervous system. So that to me would be something that maybe there will be a breakthrough in understanding how viruses and microbes and bacteria interact. And
yeah, in fact, we've done some studies in Parkinson's patients, and one of the problems with phage, and this might you know, take us down a direction now that's going to be difficult to get out of, but the problem with phage is they're so individualized. They're so diverse. There are tens of millions of species of phage. Tens of millions of species of phage. And they're so diverse that very few people share any phage. Now, this might seem extraordinary, but you know, you are full of phage. We have as many phage in our gut as we have bacteria, and I am full of phage. And while we might share certain bacteria, at least at the level of species, you know, you might have, again, Bacteroides intestinalis, and I have Bacteroides intestinalis. So we could imagine that they're doing the same things in both of us. But it is highly likely that I have thousands of phage and you have thousands of phage, and we share none. And so then it's very difficult to do comparative studies. You know, we can look at the virome in a cohort of people with Parkinson's and the virome in a cohort of healthy controls. But if everybody is different from everybody else, you end up with no grounds for comparison. While if we saw, for example, that, you know, most healthy people have Bacteroides intestinalis and Parkinson's sufferers are lacking that bacteria, and we could start to speculate, form hypotheses, test those hypotheses. But with phage, it's almost, you can't get a grip on it almost as to where to start, because the level of diversity is so high. And so what we need is much better understanding of which phage affect which bacteria, what the phages actually do to the bacteria they infect. But there are so many of them. And you know, the 99.9999, keep going with nines for a while, % of viruses have never been grown in a lab, have never been studied, have never been isolated, and we only see them as sequences when we do metagenomic sequencing of big fecal samples and things like that. And we see these signatures that we know are viruses in there, but we can never isolate them. So if you want a real challenging field to move into as a young scientist, I'd say the virome is a really challenging field, but extraordinarily interesting and rewarding, I think, if you can get into it.
Yeah, it seems to be a daunting task, and listening to you, it sort of almost explains why there's not more publications on this topic. Because I mean, to get into that, you know, it's a whole galaxy or several galaxies of things that you know have to sort of add to your.
It really is, but there are straws in the wind. There was a paper came out this week. We're talking in kind of mid-February of 2022, and there's a paper came out in Cell House Microbe this week, in which a Spanish group claimed, I don't want to be too pejorative about it, but that viruses play a role in executive function in humans, mice, and flies, fruit flies, Drosophila. And they did some experiments which were the kind of data mining type experiments, looking for evidence of different type of viruses in people. and then comparing that to their executive function. But some of them were actually interventions, where in mice they did an FMT and showed that viruses were transferred and this kind of executive function ability. And in fruit flies, they did another experiment involving phage. Now, I'd have to say, it's a straw in the wind. I'm not convinced by it, but I'm excited by it. And I want to see where that leads. And with a colleague, I know that you would know very well, John Cryan and Ted Dinan here in UCC, who are gut-brain axis scientists who look at the role of bacteria in the functioning of the brain. We are doing virome studies. And again, we have some very, very interesting preliminary results that I hope we'll be publishing soon. That does convince me the virome really does have a role to play in, at least in that aspect of the bacteriome or the microbiome function. And I believe it will also play a role in things like colitis and inflammation. The immune system sees these viruses. We get antibodies against them. The immune system samples them, takes them into the body. So they're definitely there, and we're aware of them. And I think they will have a role in different disease states and in health.
You mentioned fecal microbial transplant as a term. So I want to come back to this because I think I remember you giving a talk or a presentation on this topic that we always think it's just transferring the bacteria, but I've not heard a single person other than in your talk that at the same time we are obviously transferring viruses, and that this co-transfer of viruses or pairs of bacteria with their unique virus may actually be important for the success of a transplant. Could you explain that?
Well, we looked at a study with our colleagues in Finland, Reetta Satakori, who they had done a study of patients who'd received fecal microbiota transfers from donors, healthy donors, and these were patients with suffering from Clostridium difficile associated diarrhea, or CDI, Clostridium difficile infection. And that's a recurring infection, as you well know. Somebody gets treated with antibiotics, their bacteriomes suffer damage. This bacterium, C. diff, grows through that background then and produces toxins and causes problems. And of course, you have to treat it with more antibiotics, and that causes further damage to the microbiome. So it can be a recurring disease. It can actually be fatal in a percentage of patients. And what has been shown is that when you do a fecal microbiota transplant, so you just take feces essentially from a healthy donor and you put them into the recipient, there's over a 90% success rate, cure rate. And so what we looked to see, Reetta and her colleagues had looked to see what bacteria were transferred and did they engraft in the recipients and were they still there up to a year later. We followed up then by looking at were the phage present. And in fact, they were. These bacterial viruses were from the donor were still present in the recipient a year after the transplant event. And I'm really reaching now, but there haven't been a lot of live bacterial therapeutics from the microbiome that have worked in other conditions. But FMT is virtually the only one where the viruses are transferred along with the bacteria. And I hope there's something in that. There's a message in that maybe when we're transplanting bacteria from one individual to another, or growing them in a factory and producing probiotics or whatever we're doing with them, that maybe once again we need to think about, do they need their viral partners in order to give their full functionality? These bacterial viruses, they engage with the bacterium at their surface, of course, because they have to bind to the surface, inject their DNA or RNA, and then initiate an infection. But it's the bacterial surface that also engages with us and our immune system. So you've got the phage and us both encounter bacteria through their surfaces. And what we've shown recently, and lots of people have shown this isn't unique to us, is that if you grow a bacterium in the presence of this bacteriophage, many bacteria at least, they will kind of shuffle the proteins they exhibit on their surface because they want to give themselves a fighting chance at evading a phage if it comes along and can bind to one of its surface structures. So some members of the population will always have that one switched off, while others will have it switched on, but then they'll have a different one switched off and a different one switched on. And you can have this kind of constellation of different surfaces, even on a single bacterial strain, a single bacterial culture. And I hypothesize that this kaleidoscope of surface structures will also have a very significant impact on how the immune system sees those bacteria, how other bacteria see those bacteria, and therefore on the functionality of those bacteria. But take away the phage, and you're taking a really significant player out of that interaction. Again, hypothesis, not proven.
Yeah, I mean, that story, you know, the more you explain it, the more fascinating it gets. And you know, if you're a young scientist, you almost say, "Oh, I mean, this is hopeless," or maybe it's a problem for us older investigators, as I mean, this is so complicated, we're never going to understand this.
Yeah, yeah, I think that's been, and it was my reaction as well when we got back. I worked on bacteriophages a long time ago. Bacteriophages cause problems in this might seem a little bit mundane, but in fermentation, in food fermentations. For example, in the cheese industry, I'm sure most people are aware that cheese is made by fermenting milk with lactic acid bacteria. You put lactic acid bacteria into milk, they ferment it, they produce acid, and the milk curdles and you make cheese. And there are factories all over the world doing this. But what factories encounter, unfortunately, all too regularly for them, is a vat suddenly just fails. So you have tens of thousands of liters of milk potentially, you've put in your bacteria, and then nothing happens. And when they look, what do they find? It's phage. Bacterial viruses have infected the vat and just wiped out the bacteria. And then you have to destroy all that milk. You have to do a deep clean on the factory, refill with milk, cross your fingers and hope that no phage have got back into the next fermentation. And so I worked with people like Todd Kleinhammer in the US and Charlie Daly and Ger Fitzgerald here in Ireland on trying to understand that and to generate new versions of these cheese-making strains which were more resistant to bacteria. So I worked on phage like that a long time ago. Then, of course, switched to the microbiome over years. And then when it occurred to me, or I saw some preliminary papers from other labs, you know, maybe the viruses in the gut are worth studying. I was daunted when I started to realize that there are thousands of them. And the diversity is extraordinary. I mean, bacteriophages can have single-stranded RNA genomes, double-stranded RNA genomes, single-stranded DNA genomes, double-stranded DNA genomes. They can use alternate genetic codes. They can be as small as 3 kb, just 3,000 bases. They can be over a megabase. They can be 1 million bases. They can have genomes that are bigger than the smallest bacterial genomes. And they can have genomes that are tiny, the smallest genomes on Earth. The very first genome ever fully sequenced was an RNA phage, and again, another Nobel Prize going there. And so it was daunting. But the more and more we dip our toes into it, we're just getting sucked in more and more. It's just such a fascinating universe of biological diversity, and we're only at the tip of the iceberg. We really only study the kind of bacteriophages that have been grown in labs. And that's not even again, 0.00, and keep going with noughts for a while, 1% of the viruses that are really out there.
I mean, you mentioned two things. One is this incredible diversity, but also this amazing inter-individual difference. How does each person get this sort of fingerprint or this phenotype of their virome?
Great question. We obviously get a lot of them from our mothers, indifferently, of course, depending on the mode of birth and whether you're breastfed and things like that. But I love the Dutch scientist, Bas Beyering, and he said, now I'm paraphrasing, well, I'm kind of paraphrasing because he said it in Dutch, everything is everywhere, but the environment selects. So his idea was almost that you encounter almost everything, but then the specific environment selects out the ones that can survive in that environment, that can thrive in that environment. So I think we encounter bacteriophages all the time. If I spent some time with you, living with you, perhaps sharing food with you, sharing an environment with you, I'm sure I would encounter your phages, but my environment would potentially just not select them because they don't have an appropriate target, or I already have phages doing whatever your phages could do. And so my environment is already selected out. So I think a baby is, you know, an unformed environment, if you like. And as the baby grows, they encounter phage, they select out the ones that they need or want or that can thrive. And by that way, they accumulate their own stable, incredibly complex ecosystem. But once that's formed, it might be difficult for a phage from someone else to invade that. And so that's why we retain this incredible uniqueness in the face of all this diversity.
So, I mean, the question is, you know, who comes first? Is it like, you know, in an infant, is it their unique, you know, bacterium that, you know, is shaped by the mother that then creates an environment that allows or attracts certain viruses? Or is it the other way around? I mean, do the viruses determine to a certain degree what kind of bacteria can assemble?
Great questions again, and something that we're very keen on studying in the lab, you know, to see, do phage play a very significant role in the assembly of a complex culture from first principles? We do see, for example, that the viromes in infants who are born by C-section and infants who are born vaginally are different. But again, everybody's a bit different, but their bacteriums are different. So, you know, bacteriophages in one sense are kind of biomarkers of the bacteria that are present. A phage cannot be present in you unless it has a sensitive bacterium. It just simply cannot. It will be washed out and be gone within a matter of hours. So the presence of a phage tells you there's a sensitive bacterium in there. And so if the bacteria are different, phage de facto must be different. So we don't know. I would think that except for laboratory environments where we are very careful to transfer bacteria in a sterile way, or, you know, that when we transfer a bacterium from one test tube to another, that no other bacteria get in and that no phage get in, that's an extraordinary circumstance that has only existed in labs for the last 100 years. I'd say in almost every other setting since evolution began, no bacterium arrives anywhere without some phage around it already. And I think they probably are acquired simultaneously rather than sequentially.
Yeah, so I'm sort of coming to the end of this conversation, which has gone on for a long time, but if you had to make a prediction, so we already see a lot of commercialization of based on the bacteria testing. Seems like every patient I see comes in with a test result report and wants to know, you know, what this means. It's sort of an amazing phenomenon that the whole industry makes money on this, even though neither the physicians nor the patients really understand the results they're getting. But could you envision in 10 years from now that viruses, I mean, first of all, that we'll have a better understanding of what these you know, these relative abundances really mean in terms of prediction. But do you think viruses or patterns of viruses will become part of this?
I can't see it right now. Maybe that's just a lack of imagination. I mean, as a scientist, I'm always supposed to say, oh yes, the research I'm doing is going to lead to enormous breakthroughs in scientific or in medicine or in food or in agriculture or something. And of course, I hope they do. But I think, you know, we're so far behind in the virome. I think right now, if I were to sequence anyone's gut microbiome and show them their bacterium patterns, nobody could really make sense of it. You could probably tell if it was a really, really bad, you know, damaged bacterium, if someone had just had antibiotics and they were overwhelmed with some particular enterobacteriaceae or something. You could probably say, yeah, that doesn't look like a great bacterium. With the virome, maybe we could do the same, say, that doesn't look like a great one, but I certainly couldn't do any diagnostics. I couldn't tell you if it was a bad one yet, but we're about a decade, I think, behind the bacterium scientists. So I hope they get there, and then I hope we would follow in. because the information is certainly much more granular. You know, there's far more information, I think, in the phages about what's going on in the gut than you might get from the bacteria. But even then, it will come down to, you know, where are you sampling? And if you're talking only about fecal samples, then what are you really learning about the upper GI tract or about, you're getting a very, very blurred picture through a very opaque lens, I think, when you're looking like that. So no, we're not there yet.
I mean, there's obviously have been breakthroughs, phenomenal breakthroughs that have saved probably millions of people, these RNA-based vaccines. People wouldn't have thought this like 10, 20 years ago that this could happen. So could you see a possibility that we treat, early Parkinson's disease with some of these vaccines targeted at certain viruses?
Yeah, I mean, it's maybe in a different way to vaccines, but what we could potentially do, of course, is engineer these viruses to deliver genetic messages to our bacteria. Remember, like the Moderna virus or the Moderna vaccine essentially is delivering a message, delivering an RNA message to our cells to produce the spike protein and develop immunity. But what if we could say, well, you know, you've lots of Bacteroides in you. What I'll do is I'll deliver a message to them to tell them to do something that the bacteriologists have learned the Bacteroides needs to do for good health. Could we do the same thing? Send in messages through these viral partners and rewire your bacteriome, not completely, junk it and try and put a different one in like with FMT, but simply reprogram your existing microbiome. And that's something I'd be really fascinated to see. And I know some people have done that. They have, delivered genes to bacteria in the gut by using engineered viruses and engineered bacteriophages. So fingers crossed. I mean, what I always say to people is, I'll do my bit, and then I hope really, really bright people come along and take advantage of what we're doing and come up with fantastic ideas and therapies and modalities that will, really impact on people. There's a lot of very bright people out there. So again, the next generation of bright, young microbiome scientists, think about the virome.
So thanks, Colin, for this fascinating conversation. I think we've come to an end. Definitely one of the more fascinating podcast communications that I've had. So I hope our audience finds this equally fascinating and in an area that I would say 99% of listeners will have heard very little about. So thanks for making yourself available to this. And I'm looking forward to talk more in person in the future.
Thanks, Emran. I enjoy taking part. Thank you.
Take care. Bye-bye.